Bismillahirrahmanirrahim. Assalamu alaikum, dear students. How are you all? I hope you all are fine and doing well job at your home and you are studying well with Pakistan International School Types virtual learning classes. Dear students, this is class of biology and this is for the boys and girls of second year. In this lecture, we will see chapter reproduction, which we have started before from the previous topics. We have also discussed some topics, and today we will see in this chapter or in this lecture of this chapter reproductive systems disorders, its treatment, and some STDs. So let's get started. Disorders of reproductive system. As we have seen before in the previous chapters, there are reproductive processes which are going on in male and female, and these reproductive systems are for the production of um, like egg cells and sperm cells. Now we will see if there is some problem in the reproductive system. So, what is the reason for that and what is the effect of that? So, the main that we can say disorder, that is infertility. Infertility actually that is a disease of the reproductive system and which there is a failure to achieve pregnancy after 12 months. So, actually, this infertility is not defined precisely because there are varying degrees of infertility, and useful working definition is the failure to achieve pregnancy. So, its prevalence of what we can say its prevalence in the universe in this world, that is 40% male infertility and 40% female infertility. That reasons are known but some are unexplained in both of the genders, that 10%. So let's talk about male infertility. So, male infertility means male is unable to like having the fertilization process. There are lots of causes of male infertility which are known in them. We are having some of them here, we will discuss. So, that is azoospermia and oligospermia, sperm deformities, and autoimmune disorders. So, first of all, we will talk about azoospermia. So, azoospermia, that is a medical condition um, of a man whose semen contain no sperms. So, it means there is no sperm in the semen, or there is no production of sperms. It may be caused if sperm ducts are blocked due to infections or injuries, or blockage may be congenital or that can be by birth. It is associated with infertility in humans, as spermia affects about 1% of the male population. So that is also the cause of infertility. If we just talk about oligospermia, so it like decreased number of sperm count. You can see here in the diagram normal sperm counts and then oligospermia, less sperm count in the semen. So decreased number of sperm in the ejaculate, that is less than 20 million sperm per milliliter, or 15, that can be less than this one. which is further subdivided depending on the number of sperm into mild, then moderate, and severe. If that is like 10 to 15 million sperm per liter, so that is the mild oligospermia. If 15 to 10 million sperms per milliliter, that is moderate. And if that is severe oligospermia, so that is less than 5 million sperms per milliliter, and that is one of the major cause of infertility in men. Sperm deformity is the other reason of infertility, that the normal sperm, if you see in the diagram, there is an oval head, the first one that is normal, if you see the first two. So, and with the long tail. But in abnormal sperm have head or tail defects. So there is the changes in shape of sperms. So sperms are deformates, their structures are changed, so such as large or misshapen head, or a crooked or double tail. You can see in the diagram here double tails. Okay, so large head, double heads, long heads, like acrosome, long acrosome, curly acrosome, and like we can see the curly, that is the ring. And these are the deformities by which the sperms are formed, are produced, but they cannot reach to the female reproductive tract at the proper timings, and at the proper way. They cannot move further. So these defects might affect the ability of the sperm to reach and penetrate an egg. Because here the acrosome, the first part of the head, that is very very important regarding this penetration. So if that is defected, so it means there is um, inability to to penetrate and to fertilize the egg. Autoimmune disorder, in some individuals, the infertility is probably due to autoimmune response by the male to its own sperms. Like antibodies are produced in the bodies which recognize the sperms as antigens of any parasite or outsider invaders. They can recognize the sperms as outside invaders, so they attack on the sperms, and then they reduce the sperm count on daily basis. Now we will see female infertility. So female infertility, there are lots of causes of female infertility, like includes the problems with ovulation, damage to fallopian tubes and uterus. There is a problem in the cervix, and age can contribute to infertility. That is one of the major cause in women. So failure to ovulate, if we see this process, we have seen in the previous lecture also that hypothalamus is responsible for production of gonadotropins, or gonadotropin releasing hormones that affect or stimulate pituitary gland, and pituitary gland releases luteinizing hormone and follicle stimulating hormone. These are two very very important hormones, and these hormones affect ovary here. And ovary then is gets stimulated and starts to release progesterone and estradiol, that is also called estrogen. So that these two hormones are released from ovary, and this release is stimulated by LH and FS. So here can be the problem when hypothalamus releases less gonadotropin releasing hormone, or that is unable to stimulate a pituitary gland, or this pituitary gland that is affected or fail to produce hormones which are normally follicle stimulating hormone and luteinizing hormone. So with the result that either no follicle development can be done, because follicle stimulating hormone produces what it does, it helps in the follicle maturation. So follicle maturation will not be done, or luteinizing hormone, if it is lacked or it is not produced by pituitary gland, so there will be no release of egg, means no ovulation. So this is the reason that egg cannot be ovulate from this, and then it cannot be fertilized. So that is due to the production of less production or lacking of FSH or LH. We know that by luteinizing hormone, LH surge, like highly increased LH level, just ovulate the egg. So if that will not be produced, so egg will not ovulate and it will not fertilize. So that is the major reason for infertility in females. Now you'll see blocked fallopian tube. Fallopian tubes are these tubes which are uh, present or connected with the ovary, uh, and when the ovary ovulate the egg, they receives the egg, and the egg moves on towards the uterus, and these are the sites where fertilization occur. So if these fallopian tubes are blocked, so this is one of the major reason to um, for an infertile women. So blocked tubes may be due to the scar tissues caused by pelvic inflammatory disease, some infections, some like accidents, or some trauma can just get like a block this fallopian tube, and the sperms cannot reach here in the fallopian tube to the egg, and egg cannot be fertilized. So endometriosis is also one of the reason in which the fragments of uterine lining travel to the fallopian tube and it blocks the fallopian tube and can lead to scarring. And here you can see at this point, uh, like both of the fallopian tube can be blocked by endometriosis or any other reason. So next one we will see uterus damage. So in that we can see that fibroids, like um, fibroids or non-cancerous tumors, they are developed in the ovaries. So uterine fibroid is a leiomyoma. It means they are benign and they are non-cancerous tumors, and they are the extensions of the smooth muscles that originates from the smooth muscle layer of myometrium of the uterus. From inside, we can see here they developed outside of this one. So they can block the fallopian tube, they can block the passage to the uterus, and if there are lots of fibroids, so uterine can be damaged, structure can be damaged, and there is difficult to implant. And first fertilization, if fertilization can be also difficult to implant the egg cell here. Next, we'll see cervical mucus damage. If we remember, we have just studied that in the previous lectures, that this region is called cervical cervix, and this cervix is having the major role to produce mucus. So that mucus is thin, and it gets more thinner and crystalline when the sperms are discharged here. So that helps to to flow sperms in the cervix, and sperms can swim through it more easily. But with any type of problem, if the mucus get thicker, and sticky. So that is harder to conceive because sperms cannot enter here into the uterus more easily. So that can be difficult. So that can be blocked. That can be blocked, or that can be sticky area. So the sperms cannot enter like into the uterus. So next is we can see the endometriosis. Endometriosis is the presence of functional endometrium, like in which the egg is implanted, the inner layer, but the glands and stroma or the endometrium. What happened? They gets start to develop anywhere in the reproductive system. So we can see here in the diagram, here it can be here, anywhere into the uterus, outside the ovary, fallopian tube, or upper on the uterus if they are developed. So endometriotic lesions are there. So oviducts can be blocked, uterus pathway can be blocked, as well as there is harder to implantation of the egg, because egg is implanted in this membrane. If anywhere endometriosis can be developed, so egg can be implant anywhere, so that can be the problem for infertility. Next, we will see treatment of infertility, in which the major is a surgical, but hormonal therapies can also be given to male and female. But the now the developing technique in that is in vitro fertilization, which we'll talk about right now in detail. That is also called IVF. So, IVF or in vitro fertilization is a process by which an egg is fertilized by sperm outside of the body, that is called in vitro. So, it is a major treatment protocol for infertility. Actually, in vitro comes from the Latin word, it means in glass. So, that is used for early biological treatment for lots of cultivation of the tissues and like biotechnology in the plants and animal cells. But right now they are they are contained in the containers like beakers, test tubes, petri dishes in the glass. So that's why it is called in vitro fertilization. So due to just growing up, or this technique is done in the test tube, so that's why it's called test tube babies. Test tube baby technique. So in that, the ovaries are stimulated by a combination of fertility medication, and then one of the more oocytes are aspired from ovarian follicles, and they are fertilized in the lab with the sperms, and there's the embryos get transferred into the uterine cavity again. So let's talk about its procedure in detail. So first of all, the female gets through the ovarian stimulation, then egg retrieval is done, then sperm collection is done from the male, fertilization is done in the lab, and then embryo culture is done in the lab, and then embryo again transferred into the uterus. So these are 6 steps which are done in in vitro fertilization. So there is ovarian stimulation, the first one, in which both male and female partners that goes to the fitness test and checkups, and injectable medicines are given to the female for an average of 10 to 12 days, and a women ovaries to produce multiple eggs, like not only one follicle in the normal female. That follicle stimulating hormones FSH, as we know, and LH are given to the female body to produce more developed follicles in which egg cells are there. To ovarian response to the medications are monitored, and ultrasounds by some medications they are adjusted according to the need. Then number 2 step we will do here. Okay, in step number 2, that is egg retrieval. Like egg is now the follicles are removed and eggs are collected from the follicles by sucking out the fluid contents of mature follicles with a fine hollow needle. One needle is penetrated into the sorry, not into the uterus, but here is the ovaries. These are the ovaries. So needle is penetrated into the ovaries, and before ovulation the follicles are just removed out from this thin needle, the whole fluid in which there are lots of egg cells are present with follicles and follicles fluid. So with this, that is done by general anesthesia, and that the female must does not get any type of pain. After this one, the next process is sperm collection. In the sperm collection, at the same time, semens are prepared for fertilization by removing inactive cells. The sperms or semen is taken from the male, and there is the process that is called sperm washing, and from some washing with certain type of fluids and chemicals. So you'll get the correct numbers of or uh, defective sperms are just removed from that process, and the healthy sperms are taken out, and then semen is provided by sperm donor, like from the couple. Then co-incubation and fertilization in the laboratory, it is done like after egg and sperms has been analyzed and prepared, and the eggs are then inseminated like in a petri dish, in a test tube. There's a sperms or eggs are incubated together, and cultural medium that is given some type of nutrition to develop and actual fertilization. And during this co-incubation it should be 1 to 4 hours to higher pregnancy rates. They are just for of like for a healthy fertilization, they are given some time and monitored as well. Then in embryo culture, that embryo is cultured from 3 to 5 days until they have reached optimal growth after fertilization. So embryo culture are done until cleavage stage, 2 to 4 days after co-incubation, or blastocyst stage, because at blastocyst stage these egg cells are going to be implanted into the uterus. So or 3 to 4 or 2 to 4 or 5 to 6 days are given for after incubation, just to develop the egg cells into like a blastocyst stage. Then there's embryo culture until the blastocyst confers a significant increase in the live growth for embryo transfer. So now then when the embryo will be transferred into the uterus of the same female from which the follicles are taken out, so they are now there is high increase rate of implantation. Then embryo transfer is done. 1 to 3 embryos are transferred into the uterus 3 to 5 days after egg retrieval, and then that will be monitored. Ultrasound and proper monitoring is done. And after embryo transfer, 3 to 5 days of pelvic rest is recommended for the female. And then the test of pregnancy will again be done after 12 days to whether the egg is implanted or not. So usually test tube babies are twins, because 2 to 3 or 5 follicles are, or developed eggs are just implanted into the uterus. So this is in vitro fertilization process. You can see here Number one, egg production. Number two, egg retrieval from the ovary. Then egg sperms, and there's a co-incubation. For and sorry, then sperm samples provided, then co-incubation and fertilized eggs again just passed on into the uterus. So there can be another problem of this infertility. Fertilization is done, pregnancy is done, so pregnancy can be terminated um, before viability. That can be said as miscarriage or abortion. If it is spontaneous, so that is said to be miscarriage. If it is um, we can say the medically or mechanically that is done, that is called abortion, induced abortion, that can be said to be. And but that is done before the embryo gets the weighing of 500 grams or less. So 15 to 20% of pregnancies total reproductive losses are much higher if one considers losses that occur prior to clinical recognition. So there can be lots of some major causes for that. And so in that, so when the embryo implant near the cervix, that is also one of the reason, near cervix here near cervix, so there can be the problem, and it can lead to miscarriage because it can like move out very easily. Number two, that can be normal position placenta. But sometimes placenta can tear up and uterine wall accomplished by hemorrhage, and that can lead to miscarriage, and that can also be said to premature birth. But so premature birth, actually, if the live birth before the normal time is called EDD, expected date of delivery. But if it is done before that, so that is premature birth. It is not miscarriage because all of the organs are developed of that fetus. So let's talk about sexually transmitted diseases, in which that is also called STDs. We have just seen it before in the previous classes also, but take a review of that. The first one is gonorrhea. Gonorrhea is contagious, highly contagious sexually transmitted disease, and that is caused by Neisseria gonorrhea, that is uh, infection of the urethra, cervix, and anus, throat, and eyes, like the fluids from that is a release, and that can be the cause of gonorrhea. And then and that can infect the bloodstream, as well as it can cause fever, joints pain, and skin lesions. It can affect both male and female, and the bacteria that's found in the discharge of penis and vaginal fluid, and infect men and women, both of them. Syphilis, if we talk about, that is also sexually transmitted disease caused by bacteria, Treponema pallidum, and it is spread by sexual contact. And there can be also the painless sores, lesions, and that can cause on genitals, rectums, and mouth, from which the discharge can like produced. Then we will see HIV, or highly uh, a worldwide sexually transmitted disease and highly vulnerable, that is AIDS. And that is called as human immunodeficiency virus, that is caused by this virus, and that is called acquired immunodeficiency syndrome. The name is telling you that it reduces the immune system. Deficiency of immune system is done, so T lymphocytes, which are mode of transmission, is like by the sexual transmission by the fluids of the body, as well as the blood, semen, and the fluids of the body. It is spread by a sex from an infected partner or person. So that virus HIV, that can attack on T helper cells. These T helper cells are very important, that is actually HIV virus. So that um, T cells, these T cells, they are very helpful for attacking on any type of pathogen. So that makes our immune system stronger. But if these T helper cells are damaged, because they attack on them, they just attack them with these receptors, and they will penetrate their genetic material, that is RNA. They just get into the T cells and destroy the T cells very rapidly. So the person can get to know about this disease after 10 to 15 years, when the immune system gets too much weaker, and that is immune system is like destroyed completely. So any disease can attack on that, the person will suffer from that and cannot survive from that disease, and he can die due to this disease. So, dear student, that's it for today. I hope you understood this lecture and enjoyed it. We have completed our chapter number 20. Inshallah, in the next lecture, we will start our next chapter. Wish you best of luck. Allah Hafiz.